have it in the morning, but not in the afternoon, which is what I'm really thankful for.
Right. Okay. So here we go. Make sure you do the seating chart. Make sure you click in on iClicker for attendance. Please, please, please. All right. Why did the picture go to jail? He was framed. Framed. Poor thing. Poor thing. Okay, so I'm going to start you full screen. We're going to do chapter 10 today, but I think I wrote wrong on the screen. I think I was talking and writing and wrote the wrong things, right? For this slide. So pull up this slide on how phosphofructokinase 1 is regulated. I think I was writing and then just labeled the wrong things. So when do we want to inhibit phosphofructokinase 1? So you want to inhibit when what? Energy high or low? When energy level is high, right? Okay, so if ATP indicates a high energy charge in the cell, ATP is going to be an inhibitor or a promoter. Inhibitor. And that's what we see with the red line, right? PFK1 inhibitor. I labeled them backwards in class. AMP and ADP are indicators of low energy charge. So what are those going to be? They're going to be activators. Activators. Okay. So I just labeled them wrong in class. Y'all got that? Y'all okay on that? So think about it as a feedback inhibition, right? Do you want to metabolize glucose to eat when you're making ATP if you've already got ATP hanging around? No, what do you want to do with that sugar? We're going to shuttle it off and store it and prepare for winter, right? Winter is coming, so we're going to store as much as we can. Okay. So now we're going to switch gears and we're going to go do chapter 10. Full screen. Okay, does everybody have, y'all who just came in late, everybody get one of these? I forgot as y'all were walking in. If you haven't, come get one because you're going to need it. Come get one. So it's the Citrate Pathway. So this comes straight from your book, but it's very nice to have in front of you while you're talking about all these different reactions. So what's really, really important to remember is that the citrate cycle is not an extension of glycolysis. It's very different than glycolysis. So it's all about oxidation in citrate cycle. Citrate is all about oxidation, 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 oxidation. So those are redox reactions. So we're going to really introduce redox reactions and kind of how they work. But what's really cool about the citrate cycle is that the citrate cycle is kind of this hub, right? And it's the citrate cycle that's kind of going to decide what happens in terms of energy for the cell. It links metabolism pathways like proteins, it links to fatty acids, it links a lot of different things. So it's a very central, very important hub. And so we're going to talk a lot about how it's regulated and things like that. So in order to understand the citrate cycle, we'll first talk about redox reactions, then look at your pathway. So the circle itself is the citrate cycle. Do you start in the circle? No. What's at the very top? Pyruvate. What was our substrate? What was our product of glycolysis? Pyruvate. Okay, so we ended the citrate cycle with pyruvate. We start the citrate cycle with what? Do we start it with pyruvate? We start it with acetyl-CoA. So we have to have a reaction that is going to happen before the citrate cycle happens. So that's section 2 where we talk about pyruvate dehydrogenase converting pyruvate to acetyl-CoA. Then once we have acetyl-CoA, now we can talk about the enzymatic reactions in the citrate cycle. So be very careful that you understand what's happening in the citrate cycle and what's outside of the citrate cycle. Then we'll talk about how it's regulated and then how it's kind of the hub, which is really cool. So citrate cycle's job is to capture energy, right? All of this is about energy, and it's going to do that through redox reactions. So if we look at the citrate cycle, we know that the products of glycolysis can be used in the citrate cycle, right? But that's not the only thing that the citrate cycle can use as a substrate to generate energy carrying molecules for oxidative phosphorylation. It can also get those carbon backbones from fatty acid metabolism. So if you oxidize fatty acids, you can make acetyl-CoA. You make acetyl-CoA, guess where you can go? You can go to the citrate cycle. Same thing with amino acids. So what do you notice about each of these 3 connections? They're not one way, right? So we can actually go in reverse. This is the whole thing about a low carb diet, right? That we can then say, well, I'm not getting any acetyl-CoA through glycolysis. So I'm going to go on my low carb diet. And when I do that, where am I going to get acetyl-CoA from? Fatty acid metabolism, right? So if you eat too much, you'll go make fatty acids. If you don't eat enough, you'll metabolize fatty acids, right? And the same thing is true about amino acids as well. But it is aerobic. What does that mean, aerobic? In the presence of oxygen. So if you don't have the citrate cycle, right, it could be because you're not getting enough oxygen. But regardless, you're not going to be making very much ATP. You'll make a little ATP in glycolysis, but not very much at all. And then you're going to see the reason why we have all these links is because of shared metabolites. That's the scientific way of saying that, is that we have metabolites that are shared in these different pathways. And so we can connect to many different things. So you've probably heard it. Have you heard it as Krebs? You heard it as citrate? What have you heard it as?
Mostly Krebs.
Mostly Krebs, really. So Krebs is the person. I hate when they do this. I hate it. I hate it with a passion. Like, get over yourself. You're not that special. Please name it by its function, right? You'll get the publication. It's okay, but you don't have to name pathways after yourself. So when you guys become like super famous and like these insanely awesome scientists, you're going to remember me standing up here and saying, don't name it after yourself. Name it after a function, name it after what it does, how it works, something like that. But don't be mean to future students, okay? So Krebs was the guy who discovered it. Sometimes you'll see it as the tricarboxylic acid cycle. So why is it called tricarboxylic acid cycle? Because citrate is a tricarboxylate, right? So it's really saying tricarboxylic acid and citrate is really saying the same thing. So look at your pathway. Do you find citrate on your pathway? This is what I'm going to call it. I'm going to call it citrate. Do you see citrate? Yes. In which reaction do you see citrate? It is the product of what numbered reaction? Number 1. That's why we call it the citrate cycle, because it is the first product of the cycle, right? Okay. So because it's a cycle, why do we call it a cycle? Like, what makes it a cycle? Why do we show it as a circle here instead of as a straight line pathway? Because the very last product, so go to reaction number 8. What's the very last product in reaction 8? What do you get? Oxaloacetate. Oxaloacetate is a substrate for reaction 1. So reaction 1 requires oxaloacetate and acetyl-CoA to make citrate. So this is why it is a cyclic pathway. So when we talk about the citrate cycle in general, basically what we're saying is happening is that acetyl-CoA is going to be oxidized, right? So that's our overall goal. And every time we make that circle, what are we actually gaining? Well, the way that we're going to harvest energy is by transferring electrons. It's all about the transfer of electrons from here on out. We're going to transfer 8 electrons in total per turn. Now, how many turns are we going to make per glucose molecule that we consume? 2, because we made 2 what? 2 pyruvates. So you got to kind of keep that in mind. 1 glucose is going to give you 2 turns of citrate, right? So because of these 8 electron transfers, we are going to get 3 NAD. We're going to get 1 FADH2. What are these FADH2, NADH? These are called electron carrying molecules, right? And so we're going to undergo coupled oxidation reduction reactions in order to generate them. And we'll talk about that in a little bit more detail. And then we do actually generate 1 GTP, which we say is basically equivalent to 1 ATP. In terms of if you're counting up how many ATPs do you get from this reaction, you can count a GTP as an ATP, right? Okay. So if we look at our overall chemical reaction, right, we're starting with acetyl-CoA. We have to have NAD+, we have to have FAD. These are our energy carrying molecules that are going to pick up those electrons. We have to start with a GDP to generate GTP. And then we need that inorganic phosphate to have that substrate level phosphorylation of GDP. And then we have to do this in the presence of water, right? And we're going to get acetyl-CoA. We're going to get carbon dioxide as a byproduct that we exhale, right? We'll get 3 NADH. We'll get 3 hydrogen ions, which are going to be really important. And we'll make FADH2 and a GTP. So the next chapter, we'll talk about where all these products go and how they actually generate ATP. But eventually these, you see this NADH and FADH2, those are electron carrying molecules. So if they are electron carrying molecules, where are they going to go? Electron transport chain. The electron transport chain is going to drive ATP synthase through ATP synthesis through ATP synthase, right? So all of the enzymes that are in the citrate cycle are either embedded in the inner mitochondrial membrane or they're in the matrix of the mitochondria. But basically from here on out, once you have acetyl-CoA, everything is happening either attached to the membrane, the inner mitochondrial membrane, or in the matrix. So you have to kind of keep in mind what's going on and where. Okay. So here's our kind of cycle, right? So we have acetyl-CoA that is our primary fuel source. And our waste product that we don't want is going to be carbon dioxide, right? This is why you exhale carbon dioxide and inhale O2, because you're going to need O2 to drive the citrate cycle. You're going to make that 1 GTP, and that 1 GTP is equivalent to 1 ATP, right? That's what we said before. Now, look at your FADH2. 1 FADH2 can carry 2 electrons. When that goes through the electron transport chain, it is going to generate about 1.5 ATP molecules. FADH2 and NADH enter the electron transport chain at different places. So they are going to generate different amounts of ATP through the electron transport chain. So 3 NADH, we're going to get 6 electrons for a total of 7.5 ATP. Do you see how, even though we have basically the equivalent, right? 1 molecule, 2 electrons, 1 molecule, 2 electrons. Which one are we getting more ATP from? NADH. We're going to get more from NADH, and we'll find out why later. But for a total of 10 ATP per turn of the citrate cycle. but how many times we've gone through the citrate cycle if we've metabolized one glucose molecule? 2, right? So you got to think about, we go through this twice. So we make 10 ATP from that. All right. So how does this work? Well, lots of the reactions, right, require either NAD+ or FAD in order to run those redox reactions. And this is very, very common. These are our coenzymes. So where do we get them from, and why are they called what they are called? Nicotinamide dinucleotide and flavin adenine dinucleotide is what those 2 things stand for. Now, these are really huge molecules, but a lot of times when we write these biochemical reactions, we don't draw out the whole molecule. We only show the portion of the molecule that's important for the reaction. So what do you have for the rest of the molecule? The letter R. So just remember that these are huge, right? But we're only looking at what's important. So why do we call it NAD+? Is it because overall the molecule has a charge of +1? No. We're looking simply at the nitrogen that's in this ring right here, and we have a positive on it when it is in the oxidized form. Now, this is going to pick up 2 electrons from our reductant, right? So the reductant is giving up the electrons. The oxidized substance is going to pick up the electrons. So now we have a reduced NADH that has picked up a hydrogen, right? So how do you know oil rig? Remember, oil rig. Oxidation is loss of electrons, but how else do we do this in biochemistry? We don't just look at electrons. What can you do to look at structure? Oxidation is also loss of hydrogen, gain of oxygen. So reduction is loss of oxygen, gain of hydrogen. So what happened? You can just look at the structure and see that a hydrogen was added, right? So we can say NADH is in the reduced form because it has that extra hydrogen, right? Yes? Okay. So when we talk about these different substances, right, they're derived from vitamins. Why do we call them vitamins? So currently our bodies cannot create them. That's true. How did we discover vitamins? Like, so we look at people, right, who have a particular condition. And usually there's an environmental factor that determines they're not getting a particular nutrient, right? So you have some kind of diet where you have a dietary insufficiency. And then if you supplement with this particular food source, you can rescue the phenotype, basically. So what we're going to see, especially when we are converting pyruvate to acetyl-CoA, that pyruvate dehydrogenase, when you look at it on here, it looks real simple, right? It's a huge complex, and it requires multiple cofactors in order to make it happen. And those cofactors are vitamins. Because we noticed, and we'll talk about each one and how we discovered each one, that if people didn't have these certain things in their diet, then they had certain problems. And we could fix those problems just by adding back a particular food that had these coenzymes. So we'll talk about some different coenzymes that are really important. But FAD works very similarly, except it's in a sequential reaction where you're picking up one electron at a time, right? So what you can see is if you look at the nitrogens, right? The nitrogens are where we're going to be picking up hydrogens. So if you look at first, this hydrogen has picked up one, this nitrogen has picked up one hydrogen. Ooh. And then the second nitrogen picks up another hydrogen. So this is the fully reduced form. We call the form where you only have one hydrogen picked up the semiquinone form. But basically, we just kind of show this as one reaction. So before we talk about pyruvate dehydrogenase, I want to talk a little bit more about oxidation reduction reactions and how they're important and why they're important in citrate, but kind of in general, right? So when we talk about oxidation reduction, you always have this occurring in pairs, right? So you got to go back and remember, like Chem 20, what is it? 206. You do that in, right? You do that? No? Uh-oh. 106, not 206, 106. 106. It's a Monday. Okay, so you always have pairs, right? So if you had Fe2 plus, what's your pair? Fe3 plus, and then we're going to also have one electron, right? But do electrons exist like that? No, they don't. So usually what happens is you have a pair. So let's say copper is our other pair. So I have Cu plus and Cu2 plus one electron, right? Can I pair these two up and make one reaction that is going to give me an oxidation reduction reaction? Absolutely. And this is how it works. You make that electron, let me extend the page, the intermediate, right? See if I can make it fit. So if we start with Fe3 plus here, right? So Fe3 plus one electron goes to Fe2 plus. So now what am I going to put on the bottom for copper? Cu plus, right, goes to Cu2 plus one electron, right? So what's reducing, what's oxidizing? Which one's which? The first one's reduction, so you're reducing Fe2 plus. Second one is plus, right? So then what do you get if you combine these reactions? Fe3 plus Cu plus yields, and you really should be going in both directions, Fe2 plus Cu2 plus, right? This is our coupled reaction. So we don't actually have any free electrons floating around. So you're going to see this a lot of times in these metals as cofactors in a lot of these enzymes, because they allow for this transfer of electrons. So the next question is, why? Why do these electrons move?
You said reducing Fe2 plus, would it be 3 plus?
3 plus, sorry, 3 plus. That's terrible. 3 plus, thank you. 3 plus. All right, so why do the electrons move, right? What causes those electrons to move? Anybody? Anybody read the first section? So why do electrons move? The standard reduction potential . So E not prime, right? Same conditions as all of our standard conditions in the past. But right, we list the standard reduction potential in volts. So if you look in your book, you have a table in your book of all the different redox reactions that occur inside of the cell, and you've given standard reduction potentials for each of the individual substances, for each of the reactions. So you'll see that we have set 1 to 0. Let's see if I can pull it up. Let's see real quick. Oop. Okay. Where? We'll go down here. I want here. So if you look at the table, All right, I guess I need to go to the next section. Come on. So here. All right. So if we look here, okay, here's all of our standard reduction potentials. So if we look, we've basically said hydrogen is going to be our 0. If you have something that has a standard reduction potential that is higher than that of hydrogen, then we're going to say it's a strong oxidant. What is the strongest oxidant? Oxygen. So when you look at the electron transport chain, right, we're going to track these electrons that are picked up by NAD plus and FAD. Those we're going to track through the citrate cycle, through the electron transport chain. And the very last place that is going to pick up electrons, the very last substance that's going to pick up electrons is, take a guess, oxygen. It has the strongest oxidation potential, right? So the way that these reactions are set up is that we take things that are able to give up the electrons and pass them to things that have higher and higher and higher and higher reduction potentials, right? So you start with something that doesn't have a very high potential at all. It's actually negative, right? And we're going to keep passing electrons to things that want them more and more and more and more. And that's the reason why electrons are going to move. You're going to see that it's a very stepwise process that's going to generate the electrons needed to drive the electron transport chain. So you don't have to memorize these. You just have to understand it, right? That this is why electrons move. So don't worry about memorizing the standard potentials. Like, you know, if I need you to know that, I'll give that to you. But you have to know why they are moving. Okay, so, and how do you know if an electron is going to move? How do you know if we're going to have something that's going to move from, let's say, succinate, right? That's going to pick it up in order to go to a cytochrome. How do we know that's going to happen? You have to look at the difference in standard reduction potentials, right? So if we look at, let's say, cytochrome A3, right, it has a reduction potential of 0.35. If we look at succinate, right, this reaction has 0.03. So all we have to do is subtract the two. And if you get a positive number, then you are going to have that electron move, right? So it's kind of like delta G. If you get a negative delta G, the reaction is spontaneous. Here, if you have a difference, if you have a positive difference, that electron is going to move. Yeah. Okay. All right. So we said oxygen's our strongest one, right? Let's go back here. All right. So oxygen is going to be the very end. So we're going to have to back up. Where are we starting at? We have from glycolysis, what's our product? Pyruvate. We need to convert pyruvate into something that can be used in the citrate cycle. Yes.
That reaction.
Yeah, this one. Oh. This one. Good. Okay. Oh. I thought you had it. Okay. Okay. So we got to convert pyruvate to acetyl-CoA so that it can be used. So when we talk about pyruvate dehydrogenase, it is not just one very simple thing. It is a huge complex. And the huge complex actually contains 3 different enzymes. So it has a pyruvate dehydrogenase. If something is a dehydrogenase, Do we have an oxidation reduction reaction going on? Absolutely, right? We're taking away hydrogens. We have oxidation reduction. Then we have a trans-acetylase, and then we have another dehydrogenase. So it's absolutely redox reactions that are occurring. So just for the conversion of pyruvate to acetyl-CoA, we have to have 5 different coenzymes. We talked about NAD+, we talked about FAD. Now, we're going to generate acetyl-CoA. So we have to stick an acetyl group onto coenzyme A. So coenzyme A has to be a coenzyme. Then we're going to have thymine pyrophosphate, abbreviated TPP. And then the last one is alpha lipoic acid. But some books and some people call it lipoamide. So you might see it either way. So just realize it's the same thing. Okay, so all of these are actually derived from vitamins. So all of our coenzymes here, these are derived, D-E-R-I-V-E-D, from vitamins. So we have evolved over time that we consume things that have these vitamins inside of them, right? So plants, on the other hand, plants can't do that, right? What do plants eat? Sunlight. So they don't have vitamins because they have to generate everything. So they have all the enzymes in order to make these coenzymes. We have evolutionarily lost the enzymes to create these on our own. Therefore, we have to get them from dietary supplements, right? So this is why vitamins really do work, right? Okay, so coenzyme A. Coenzyme A is a derivative of vitamin B5. So vitamin B5 is panthenoic acid, H-I-N-O, panthenoic acid. Wish I could spell, right? And so the whole point of this is to have something that will generate acetyl-CoA. So here's our whole coenzyme A right here. And so what we want to do is we want to have a reactive end. In this case, it's a thiol group that is going to actually react with an acetyl group. And this acetyl group was originally from pyruvate, right? So this is kind of later on down the line. We have a pyruvate dehydrogenase that is going to remove the hydrogen, and then we're going to be able to stick our acetyl group onto acetyl-CoA and then generate the acetyl-CoA that's necessary for the first step in the citrate cycle. Okay. So acetyl-CoA, coenzyme A, you don't need very much, but it's really easy to get in your diet. You find it in like chicken, you find it in yogurts, you find it in avocados. It's actually really high in avocados, but you get it quite easily in your diet. Yes, ma'am. Panthenoic acid is another way to say a vitamin B5, because everything had a name before it became a vitamin, right? Then once we recognize, oh, this is necessary in your diet or you're going to have some kind of disorder, then we started calling them vitamins. And then it was really easy just to give the vitamin a letter and a number rather than having all these special names, because people don't like that. But if you call it vitamin A and vitamin B and vitamin, right, people are more comfortable with that rather than the actual scientific name. So everything has an actual scientific name. You know, people are scared of dihydrogen monoxide. But what is that? Water. There was, you know, they did a petition on a college campus and you know, gave all these facts about how if you ingest too much dihydrogen monoxide, you know, it's going to have all these detrimental effects on, you know, and they actually got people to sign it. That's the sad thing. That's a really sad thing, is they got a lot of people to sign it. All right. Sidetrack. So thymine pyrophosphate. This is, have you ever heard of the disorder beriberi? Beriberi? No? So beriberi, is a disorder. The people who have this, they look very anorexic. They have cardiovascular issues, they have neurological issues, but it's all in parts of the world that rely on white polished rice, because you actually have lots of this TPP or lots of vitamin B1 really inside the bran that's over the rice, right? And so depending on how you process your rice, you can lose the bran. And if you lose the bran, like in polished white rice, then you don't get it. So you can have, this is why we have fortified rices and cereals. Have you seen that before on the brand? It's fortified, right? So all they did was add this vitamin back. That's what fortified means. If you eat brown rice, it's in there, right? You don't have to add anything back. But, you know, side note. Okay. So vitamin B1, when it's processed inside the body, it generates our thymine pyrophosphate. So what's happening here is that we actually want to add two phosphate groups, right? Pyrophosphate. Whenever you see pyro, this doesn't mean fire. What does it mean? Two phosphates. Just means two phosphates. Pyrophosphate is two phosphates hooked together. It's not a big deal. But what we want to do is we want to provide an attachment site for activated aldehydes. And that's going to be, you're going to see this in the mechanism in just a minute. But it's the functional component for the aldehyde transfer, which we'll see in just a second. Okay, last one's alpha lipoic acid. This is the same thing we said, lipoamide, right? So this actually provides a reactive disulfide. So remember when we talked about disulfides, reducing and oxidizing disulfides inside of proteins and things like that? It's also really important in our preparation to go from why am I blanking? Pyruvate in order to get to acetyl-CoA. Woo! So when you talk about the components of pyruvate dehydrogenase, we call them E1, E2, E3. It's just an easy way to refer to them. So E1 is our pyruvate dehydrogenase. E2 is dihydrolipal acetyl transferase. So if you look at these, right, here's pyruvate coming in. How many carbons do we have on pyruvate? 1, 2, 3 carbons, right? We went from a 6-carbon glucose to 2 3-carbon pyruvates, right? Yes? Okay. So in our very first reaction, we're actually going to lose one of those carbons. So that in our next reaction, we're only going to have 1, 2 carbons that are attached to the TPP. So in that very first step, we lose one of the carbons and we start producing waste of carbon dioxide, right? I think a lot of people think that that kind of happens at the end, but it doesn't. Okay. So now what's going to actually happen in the dihydrolipal acetyl transferase is that we'll not only regenerate our TPP again, but then we need to look at here. This is our acetyl. We're going to move our acetyl group to the hydrolipoamide. Okay, so we have this reactive sulfur here that can pick up that acetyl group. Then we're going to go through another step where we're going to attach that acetyl group to our coenzyme A. So you go from having a 3-carbon chain, shorten it to 2, and then attach it to coenzyme A. So it's all about transferring those 2-carbon units. So here's our entire reaction, right? So this is the entire complex. So they've highlighted them as enzyme 1, 2, and basically enzyme 3 here. So if you look, you actually have 5 different steps within the mechanism, right? So we said we start out with pyruvate, our 3-carbon. We're going to transfer 2 of those carbons to TPP. Then we transfer those 2 carbons to the lipoamide, right? We have our disulfide, it gets reduced right here. Now we have our acetyl group attached to one of the sulfurs. Now that we have it in a reactive form, we can react it with CoA, we can attach it to CoA, but then at the same time, you have to regenerate everything so that you can go through this and do it all over again, right? Just like when we looked at all those enzyme mechanisms in chapter 7, 8, no, anyway. You have to regenerate them, right? So we have to create that disulfide bond between these two sulfurs in the dihydrolipamide. So we do that by transferring electrons, right? So we convert FAD to FADH2. The FADH2 then converts NAD plus to NADH. So that's where these coenzymes come into importance into regeneration of your lipoamide. So go through and see if you can walk through the mechanism. It's a pretty important mechanism. So if you're looking at kind of the general reaction, right? And this is really important for thinking about regulation. You need pyruvate, you need CoA, and you need NADH. And what are you going to produce in the end? You're going to produce your 2 carbons, your acetyl group attached to CoA. You're going to remove one of the carbons. You're going to produce NADH and you're going to produce H+. So when we do this, you have to think about regulation. So how are we going to regulate this reaction? Because this is going to be really super important to regulate. When do you want this reaction to occur? It has a very high negative delta G value. Look at this, negative 33. So it's irreversible. So it's going to play a really big role in regulation. So when do we want pyruvate dehydrogenase to be active? Do you want it to be active when citrate has been working for a while? No, right? So you got to think about feedback inhibition, right? So what are you going to get out of the citrate cycle that is going to inhibit entry into the citrate cycle? So what's our very final end product? Well, think about end product of cellular respiration in general, like we're all the way done. ATP, right? So ATP is going to inhibit pyruvate dehydrogenase, right? That makes sense. What is a product that is made in the citrate cycle? NADH is something we make in the citrate cycle. So this is going to inhibit, right? So we have NADH, we have ATP that's going to inhibit. Acetyl-CoA. This is the exact product of the pyruvate dehydrogenase reaction. So you have an immediate, this is immediate feedback. And what I call NADH and ATP, I call these like long-term feedback. Feedback, right? You are going to make one NADH in this reaction, but really the majority of your NADH is going to come from the citrate cycle. So those NADH, the ATP, the acetyl-CoA, what kind of inhibition is that? So it's negative, right? NADH, ATP, acetyl-CoA. These are all inhibitory, but what kind of inhibitory? Like, how does it work? It's allosteric control, right? What does allosteric control mean? If you have something that allosterically regulates an enzyme, what does it do?
The end product also acts as the inhibitor.
So it's an inhibitor, right? So allosteric control means if you bind to it, you're going to induce a conformational change that is going to inhibit the activity of the enzyme, right? That's allosteric. So this is allosteric control. So NADH is actually going to compete with NAD plus for binding to the enzyme, right? Same thing with acetyl-CoA. Acetyl-CoA is going to compete with coenzyme A to associate with pyruvate dehydrogenase. So these two, NADH and acetyl-CoA, are competing with their alternate forms, basically, right? So the more of those forms you have around, then you're going to either inhibit or positively influence the pathway, whichever one's there. Now, ATP, does pyruvate dehydrogenase require ATP? Do you see ATP anywhere? Nope, does not require ATP. So this is an allosteric control. It will bind, it will induce a conformational change difference, but it doesn't compete with anything. It's just kind of like if it's present, right? So if that's allosteric, we'll call this negative allosteric control. So let's talk about positive allosteric control. What substances? I just told you all of them, actually. Well, not all of them, minus one. If NADH is a negative allosteric control, what's a positive? NAD+. If acetyl-CoA is a negative, what's the positive? Coenzyme A, CoA. Okay, if ATP is a negative allosteric control, what's going to be a positive allosteric control? ADP. Okay, there's another form of control for pyruvate dehydrogenase. The other form of control is actually a covalent modification, right? So we'll say also regulated by covalent modification. What's the most common form of covalent modification that regulates enzyme activity? Phosphorylation. So you have phosphorylation of E1, right? So phosphorylation of E1 is a negative regulator. But you got to think, if you're going to phosphorylate it, you also have to have an enzyme that dephosphorylates it. And we're going to talk about that in the towards the end of the chapter, where we talk about what phosphorylates and what dephosphorylates. The only other one that exists that is not on the list that we just wrote is you also have a positive allosteric control with Ca2+, but this is primarily in muscle cells. Phosphorylation of E1 and a minus sign. That's negative regulation. And the calcium actually promotes the dephosphorylation of E1. That's how calcium kind of works. But that's in muscle, so you don't have to know that too much. Okay. So very, very, very highly regulated point in the pathway. So two main mechanisms of regulation of pyruvate dehydrogenase are allosteric control and covalent modification through phosphorylation, right? Okay. So y'all have heard of arsenic before, right? As a poison, right? So this is actually possibly how Napoleon Bonaparte died, possibly, maybe. But they used to use this to poison kings and queens and things like that way back when. And it actually creates a covalent bond with our reduced lipoamide. So this arsenide here bonds to the two reactive sulfur groups on the lipoamide. So now, can this go through and bind to your acetyl group? No, those sulfurs are in a covalent bond with the arsenic here. So you've basically shut down the person's ability to even enter the citrate cycle. Are you going to have serious problems? Absolutely. Absolutely. It's pretty nasty way to go. I'll pass on that, right? I'll pass. Okay. So now we can really start talking about reactions of the citrate cycle. And I almost don't want to start that yet. Let's try to do a summary. I want to kind of do a summary of our everything for pyruvate dehydrogenase, because it's like one simple step on here, but it's really not that simple, right? Okay, so for pyruvate to acetyl-CoA, are we in the citrate cycle? Number one. No. What enzyme catalyzes this reaction? Pyruvate dehydrogenase. My pen would work. Pyruvate dehydrogenase. How many subunits? 3 subunits. We call them E1, E2, E3, right? How many cofactors? Right? So what do we call these cofactors? What's another name for it? Vitamins. INS, vitamins, right? So that's something we have lost the ability, if I could spell vitamins, that we have lost the ability to create on our own. So let's talk about each of our reactions. Let's talk about E1. What happens in the very first step? We're converting pyruvate to what? We're going to release something, CO2, and what do we have left? An acetyl group, a 2-carbon acetyl that we attach to what? To what? One of the cofactors. TPP. Right? Okay, how about E2? We take our 2-carbon acetyl group, right? And we transfer it to what? Another coenzyme. What coenzyme? It's the one with the 2 sulfurs. We'll just call it lipoamide. Now we're going to make acetyl dihydrolipoamide. Okay. All right. And then in E3, what happens? What happens with our 2-carbon acetyl group? Do you see how it's just shuffling around this acetyl group? CoA, coenzyme A. Now, why do we not see NADH, NAD, and FAD? Why do we not see those here in our descriptions? Are they important cofactors? Yes. Are they important in generating the substrates for citrate? No. What are they involved in? Regeneration of the coenzymes that are required for the pyruvate dehydrogenase reaction, right? So it's kind of important to understand where the different cofactors are being used. So you could say like FAD linked to which one of these? It's linked to E3 to reoxidize, right? That's its job. The dihydrolipoamide. I kind of like science names because, like, if you can spell just the little pieces, you can put it all together. Okay, so that's FAD. What about NADH plus, right? So So FAD is linked to E3. And then FADH passes what? 2 electrons to our NAD plus to form NAD plus, I'm sorry, NADH plus H plus. Right? So it's important to know where those cofactors are working and kind of what they're doing. That make sense? So even though we were starting citrate today, we didn't actually start citrate today, right? We actually just made the substrate for the citrate reaction. Now, before you walk out the door, I am going to go home tonight and I am going to make a video for Wednesday. Okay? I'm still on the calendar, right? Let's see the calendar. That's calendar. So the question is, when are we shooting for the exam? So did I pull the exam off? What did I do? 10/10. Oh, here, I know it's here, Friday.
So it's a Friday.
So the Friday after we come back. So we'll have 2 days. We'll have the citrate again on Monday when I see y'all. We'll start oxidative phosphorylation, but I'm not going to put oxidative phosphorylation on the exam because that's not nice. I'm not doing that.
Thank you.
Yep. But then we'll finish oxidative phosphorylation the next week.
The YouTube videos from Friday didn't get posted to the playlist.
It didn't? Oh, Fridays are hard. for
Yeah, it's
me.
okay. Yeah.
Fridays are hard. for
You're
me.
good. Yeah.
Would you send me an email?
Yeah, I'll let you
Because
know.
I've got like today.
Yeah, you're
2
good.
weeks to send me an email and I will post it.
Of course, yeah.
Yes, thank you for telling me.
So just to clarify, the test is still scheduled for that next, for the
Yes.
Friday we come
So
back?
instead of actually having to show up on
Okay.
Wednesday,
Okay, we'll have
there's a video.
Okay.
I think that's probably good for everyone.
Thank you.
Do we not synthesize vitamin D? Are there some vitamins we do synthesize?
You only synthesize vitamin D if you go out in the sunshine.
So
So
we do, but that's the
because
only one?
if you don't go out in the sunshine, right, it's almost like a dietary insufficiency, except vitamin D you're not getting from your diet, you're getting from UV exposure.
The UV, what it oxidizes or reduces?
Yeah. So
I don't actually know.
whenever, but anytime you talk about a vitamin, you're always talking about if you don't get it, right, you have to get it from your external environment. You can't
You're
synthesize
saying if I
vitamin D
stay
on
in the
your own,
dark room, okay,
right? Your body all by itself can't make vitamin D.
okay.
You have to get exposed to the sun. You can't make vitamin B by yourself. You have to ingest it, right? So you're getting it in a different way, but you can't make vitamin D yourself without an external.
You make it so that you
Input.
need something.
Yeah.
Okay, all right.
Yeah, that makes sense. Okay, let me end the Zoom. Anybody on Zoom have questions? The participants are, yeah, no problem. Anybody on Zoom have questions? There was no chat. Okay, let me end this.
Do you mind if I come in at 12:15 so I can go grab lunch?